Welcome to Oslo. We are here today to celebrate the best science and the finest scientific minds. To explain more, please welcome the President of the Norwegian Academy of Science and Letters, Lise Øvreås.
Today is a great day for science and excellence in science. Welcome to the Norwegian Academy of Science and Letters and the announcement of the 2024 Kavli Prizes. Today we're honoring groundbreaking work in 3 scientific fields. Work that has provided us with brilliant new knowledge, insight and ways of thinking about astrophysics, nanoscience and neuroscience. In the words of the founder of these prizes, Fred Kavli, the largest, the smallest, and the most complex. Without any delay, let us go straight to the new winners. And so we begin with the largest. The Kavli Prize in Astrophysics for 2024 is awarded to David Charbonneau and Sarah Seeger for their groundbreaking work on the discovery and characterization of extrasolar planets and their atmospheres. The Kavli Prize in Nanoscience for 2024 is awarded to Robert Langer, Paul Alivisatos, and Chad Merkin for pioneering work integrating synthetic nanoscale materials with biological functions for medical applications. And finally, the Kavli Prize in Neuroscience is awarded to Nancy Canwisher, Vinrik Freiwald, and Doris Sau for the discovery of a highly localized and specialized system for representation of faces in the human and non-human primate neocortex.
Thank you, Lisa. From the bottom of my scientific heart, congratulations to all of this year's Kavli Prize winners. We'll get to know these scientists and their work a little bit better in just a minute. Each one of the chairs of the 3 prize juries is going to present the citation, and we'll get to meet the scientists themselves. So let's just dive in, starting with the biggest, astrophysics. The study of extrasolar planets and the groundbreaking work this year's winners have done when it comes to the discovery and characterization of exoplanets and their atmospheres. Please welcome Professor Viggo Hansteen, chair of the Kavli Astrophysics jury.
Exoplanet science has moved from the detection of the first giant planet orbiting a solar-type star outside of our planetary system to the revelation of the diversity of exoplanets, the demographics of their global properties, and the physical characterization of their atmospheres. The last decades have seen amazing developments in this field. A new category of planets, the super-Earths, were discovered, and it was realized that there's an abundance of low-mass planets. In fact, the Milky Way may contain more than 100 billion planets, with low-mass planets by far the most common. Measurements of mass and radius have allowed determination of the mean density of planets, and with that, the discrimination between gas giants and rocky planets. Furthermore, locating rocky planets in the habitable zone where liquid water may exist is now possible. Today, the characterization of exoplanet atmospheres, particularly via transit spectroscopy, is an emerging field. Sarah Seeger and David Charbonneau have pioneered methods for the detection of atomic species and planetary atmospheres and the measurement of their thermal infrared emission, setting the stage for finding the molecular fingerprints of atmospheres around both giant and rocky planets. The next big step lies in detecting molecular biosignatures, an endeavor involving significant technical challenges. Seeger and Charbonneau have led the field, developing innovative instrumentation concepts and the basis of progress for current and future exoplanet science. David Charbonneau is Fred Kavli Professor of Astronomy at Harvard. He led the discovery team for the first transit of the giant exoplanet HD 209458b, which allowed the precise determination of the planetary surface gravity and its mean density, proving that this planet is a gas giant. Charbonneau pioneered the application of space-based observatories to perform the first studies of the atmospheres of giant extrasolar planets. In 2002, he used the Hubble Space Telescope to directly determine the chemical composition of the atmosphere around a giant planet and found evidence for sodium in its atmosphere. In 2005, he used the Spitzer Space Telescope to make the first detection of a thermal infrared emission from an exoplanet. He was a member of the Kepler mission and determined the occurrence rates of exoplanets. Presently, he's actively involved in the Transiting Exoplanet Survey Satellite mission and James Webb Space Telescope observations. Charbonneau currently leads the innovative M-Earth project, aiming to detect rocky planets. Sarah Seeger is the class of 1941 professor of planetary science, a professor of aeronautics and astronautics and of physics at MIT. She pioneered the theoretical study of planetary atmospheres and predicted the presence of atomic and molecular species detectable by transit spectroscopy, most notably the alkali gases. Seeger made outstanding contributions to our understanding of planets with masses below that of Neptune, finding that higher mass variants are dominated by hydrogen and helium, while smaller mass ones share the properties of rocky bodies. Seeger provided new concepts for our understanding of the habitable zone, where liquid water can exist, and then established the importance of a variety of biomarkers. She was among the first to recognize the importance of a star shade as a free-flying external occulter, to block the light of the parental star and to allow the detection and characterization of a second Earth around a solar analog.
Thank you, Viggo. Genuinely one of the most exciting areas in modern science. We've gone from 9 planets to thousands in just 3 decades. And here are the 2 laureates in astrophysics, David Charbonneau and Sarah Seeger.
For all the exciting discoveries over the past 25 years in discovering exoplanets and characterizing them, I think that the greatest discoveries still lie ahead of us. NASA and other space agencies around the world are now making plans to build extremely powerful observatories that are devoted to studying the atmospheres of Earth-like planets and really understanding, are they like the Earth, are they different, and is there actually life on those planets? It's perfectly reasonable to hope that in the coming 10 or 15 years, we might really know the answer to this question that has been with us for millennia.
I'm proud to be leading the international consortium we call the Morningstar Missions to Venus. We aim for a series of private-public partnerships to send a series of focused astrobiological missions to Venus to search for signs of life or even life itself. To motivate all of this, we are carrying out laboratory experiments in sulfuric acid, demonstrating that biological molecules, some of ours and some different potential ones, are actually stable in sulfuric acid.
Let's move on to nanoscience and the pioneering work of 3 scientists in integrating synthetic nanoscale materials with biological function for medical applications. Please welcome Bodil Holst, chair of the Kavli Nanoscience jury.
One of the most virtuous goals of science is that of preserving health and saving lives. The 2024 Kavli Prize in Nanoscience honors 3 scientists whose breakthroughs have combined nanostructured synthetic materials with biologically active molecules. They have pioneered their use in therapeutics, vaccines, bioimaging, and diagnostics, contributing foundationally to the field of nanomedicine. In addition, the 3 laureates, Robert Langer, Paul Alivisatos, and Chad Merkin, have contributed to the societal impact of their research by founding companies to translate the fundamental science into biomedical applications. Robert Langer had the breakthrough idea that a material could be nano-engineered for the controlled release of therapeutic biomolecules. While Langer's ideas were initially received with skepticism, he showed that polymers could be synthesized to permit the regular flow of drug molecules through channels in the material. This idea has had profound impact on the development of controlled drug delivery systems. Langer's work has had immense clinical impact for the treatment of diseases such as an aggressive brain cancer, prostate cancer, and schizophrenia. Langer also showed, as far back as 1979, that tiny particles, in this case containing protein antigens, could be used for vaccination. Paul Alivisatos pioneered the use of semiconductor nanocrystals for biological imaging by tailoring them through surface functionalization. Semiconductor nanocrystals, or quantum dots, are nanoparticles that possess bright, size-dependent, light-emitting properties. Alivisatos showed that these nanoscale beacons can be used as multicolor fluorescent probes in biological imaging. Semiconductor nanocrystals are widely used today in applications such as live cell tracking, labeling, and in vivo imaging. Chad Merkin introduced the concept of the spherical nucleic acid, SNAs for short, consisting of a nanoparticle core coated with a dense layer of oriented synthetic DNA or RNA. This nanoparticle biomolecular complex gives rise to a very high binding specificity combined with a nanoparticle reporter. With SNAs, Merkin has been able to show ultra-sensitive and selective detection of proteins, DNA, and RNA. This has led to a fast, automated, point-of-care medical diagnostic system.
Thank you, Bodil. Basic science all the way to working products in medicine, highlighting the translational potential of the science right there. Let's hear from the three Nanoscience Kavli Laureates, Robert Langer, Paul Alivisatos, and Chad Merkin.
The work that we did started actually over 50 years ago, where we were trying to make small particles that could deliver large molecules like proteins or nucleic acids like RNA or DNA. And we did that to study cancer. People really felt at the time that was impossible, but we were able to, after many, many failures, get something like this to work. And over the years, not only myself, but many of my students and many other scientists and companies around the world, kept improving on these approaches and making them better and better. And that this has led to all kinds of medical products, different microspheres or nanospheres that can deliver drugs to treat cancer, to treat mental health diseases. And much more recently, it's led to delivery systems for messenger RNA that can create COVID vaccines, and people are looking at this for treating cancer vaccines and many other diseases.
We were able to demonstrate that quantum dots could be prepared and organized and coded in such a way that they could, for the first time, be used to be directed to specific locations in a biological specimen to enable improved imaging over what was possible before.
Spherical nucleic acids are a great example of the importance of nanotechnology. I think of one of the most important molecules ever discovered by scientists, ever synthesized by chemists, as DNA. If you take short snippets of DNA and rearrange them on a tiny little particle, a nanoparticle, made of anything, it will have new properties. And those properties derive from that structure that is characterized by a shell of a oligonucleotide surrounding a central particle core, a spherical nucleic acid.
Finally, let's move on from the smallest to the most complex, neuroscience, and the discovery of the system for representation of faces in human and non-human brains. Please welcome Kristine Walhovd, Chair of the Kavli Neuroscience Jury.
Recognizing faces is important for social interaction in many animals. Identifying group members is crucial for forming social relationships, and for many primates, face recognition appears to be an efficient way of differentiating between individuals. It has long been assumed that faces are treated as special by the brain. Human infants, with many other mental capacities not yet developed, seem to be born with the ability to recognize faces and may even imitate facial expressions. Following some brain injuries or deviant development, the ability to recognize faces may be deficient, despite effortless recognition of other objects. All of this hinted at the existence of a functionally specialized system for face recognition. Indeed, isolated face-selective neurons had previously been detected in non-human primates, and positron emission tomography studies also indicated selective neural foundations. However, face recognition had not been clearly localized to a specific area of the brain. Our laureates, Nancy Canwisher, Winrich Freiwald, and Doris Zhao, used functional magnetic resonance imaging, fMRI, to localize different areas in neocortex specialized for face processing. Nancy Canwisher pioneered the establishment of the Functional Region of Interest (FROI) approach to localize the fusiform face area in humans using fMRI. Canwisher was the first to develop and employ a paradigm to identify a region sensitive to faces in individual people. This finding strongly supported the idea of modular localization of cognitive function in the neocortex. The use of such functional localizers is now widespread and has been applied to domains beyond the face recognition system. Inspired by Canwisher's findings, Winrich Freiwald and Doris Zhao together used fMRI to localize similar brain areas or face patches in macaques and then went on to record from single neurons in each patch. They showed that the overwhelming majority of visually responsive neurons in the largest such region were face selective. They proceeded to outline a system of multiple face patches detailing their interconnections and functional specialization. Face recognition in the earliest patches was dependent on viewpoint, the angle from which the face was viewed, but later became viewpoint independent through a series of processing stages. Winrich Freiwald, in further work, characterized populations of cells selectively responsive to faces familiar to the viewer. Doris Zhao identified different features of the face that make up a code enabling single cells to identify faces. Together, the laureates, with their work on neocortical specialization for face recognition, have provided basic principles of neural organization which will further our understanding of recognition of objects and scenes.
Thank you, Christina. An area of basic neuroscience that reveals a fundamental part of being human. And here are the three Kavli Neuroscience laureates: Nancy Canwisher, Winrich Freiwald, and Doris Zhao.
In the early 1990s, a bunch of smart physicists figured out how to take a regular old MRI machine and get it to show not just structure, but function, where neurons are firing in the brain. And I came along a few years later and I thought, this is awesome. We're going to use this to look at the structure of the human mind. And one of the first experiments we did, we popped subjects in the scanner, we showed them faces, we showed them objects, and we found a little patch of the brain right there in everyone that responds more when you look at faces than when you look at objects. And since then, we've used similar methods to go on and chart lots of other specialized regions of the human brain, which collectively give us an initial rough sketch of the structure of the mind.
Faces allow us to recognize individuals that we know very well. In fact, these two processes, face processing and the recognition of individuals, are so closely linked that we are often not even aware that they're different. However, it turns out that it's not the general face processing network that is selective for the processing of familiar individuals, but two additional areas that we discovered in a different part of the brain. These two areas form a link between the perception of faces and the recognition of individuals that we know very well.
It was so surprising to put an electrode into a face area for the first time and find that almost all the cells responded selectively to faces. And this just opened the door to the carpenter shop. And now we could study, how the brain is actually constructing this ineffable thing called facial identity. And then figuring out what the code that the cells are actually using is, discovering that the single neurons are representing different features in this high-dimensional face space, and we could even reconstruct the face that the monkey's seeing from responses of a few hundred neurons only. It was really, really satisfying.
Well, there it is. I do hope that you've enjoyed our ceremony and the presentation of the Kavli Prize winners for 2024. Fundamental, curiosity-driven science that reframes how we understand the universe and our place in it. Thank you from all of us here at the Norwegian Academy of Science and Letters. The award ceremony will take place at the Oslo Concert Hall on Tuesday, the 3rd of September. So we will see you there.